Good afternoon, good morning, and good evening. Welcome to Mint Medical's live webinar. Today, we're going to be showcasing some of our new features that we're going to be having at the RSNA 2019 that's coming up in just about a couple of weeks. So this will give you an idea of what to expect if you have the time to come by our booth. So as before, this session will be moderated by Tobias Gottman, who is the Chief Sales Officer here at Mint Medical, as well as myself, AJ. I'm a Senior Sales Consultant. The webinar is set up in the following way. So all the participants are currently muted. That means you can hear us, but we cannot hear you. But if you have questions, please type them in. You will find a section on the GoToWebinar screen where you can type in your comments and questions. And we will look at them and we will send you an email after the webinar with the answers as a follow-up. And lastly, the recording of this webinar will also be sent to you in an email in the next days. Today's agenda is as follows. In the beginning, we're going to start with an introduction about Mint Lesion as an application. Then we're going to go into the RSNA preview for the following use cases. So the first one would be in terms of how Mint is used for structured reporting in clinical routine practice, so things like screening and staging. Then how it is used in clinical trial site reads. Then we're going to showcase our new Radiomics module. And finally, give you a spotlight on the AI features that we're currently working on. At the end of the presentation, we will give you some more details as to where you can find us at the RSNA. This entire webinar presentation will be approximately half an hour. With that, I'm handing it over to Tobias.
Thank you, AJ, and welcome everyone to our webinar. I just got an overview of who is attending this webinar, and we are very, very happy that we have more than 200 people joining live from all over the world. For those of you who just start over to learn about Mint, we will give you a short introduction. But of course, today we will focus on the RSNA 2019 showcases. A question we get asked very often is, what is the difference between Mint Lesion compared to other software solutions? And actually, the technical answer is very complex. But let me try to answer it with simple words, as I would explain it to a friend of mine who doesn't know anything about radiology, and in just using one slide. The current approach in radiology consists of 3 different non-integrated instruments. Radiologists use a PACS viewer to view the images. They use the reporting to communicate their results based on dictation. And for specific reading tasks, they also consider, or they should consider, guidelines, criteria, or classifications, which they know by heart or which they look up in booklets or somewhere else. This current approach has a lot of advantages. For example, it's quite fast, and speed and efficiency is one of the most important requirements for you as a radiologist. And this workflow also offers a huge flexibility for each and every radiologist. But there are also some disadvantages. There's a certain risk of missing information or misclassification of results, especially for unexperienced radiologists. Information are not directly reproducible, since communicated information and reporting is not linked to each other. There's a wall in between. We have a high variety of reporting styles leading to a non-optimal communication with referring physicians. And also important, the information you generate as a radiologist is not machine readable. It cannot be mined. We at Mint offer another approach for radiologists, since we combine all 3 instruments, viewer, guidelines, and reporting in one software platform, offering a cognitive assistant approach. In our approach, Mint knows about the job to be done by the radiologist, and the software guides through an assisted read applying knowledge modeling as well as machine learning technologies. The main aspects are that we provide criteria-conform reading and reporting. The read-to-report approach leads to the reproducibility of every information provided. This aspect is also quite interesting for applying AI. Imagine you have an AI-suggested lesion, and the radiologist does not agree with this suggestion. He or she will dismiss the lesion in Mint, and the report will be generated without this lesion. In addition, the system learned that the AI-suggested lesion was not approved by the radiologist, resulting in a positive feedback loop to make AI applications better and better over time. Also, reading templates are configurable and could be changed by template owners or medical champions in every institution. And last, but maybe one of the most important aspects, MINT generates labeled, longitudinally connected, machine-readable image and reporting data. We are convinced high-quality data is the future of healthcare, and MINT is delivering those data. Now, where to apply this approach? Let us start with the big picture, the landscape of imaging in cancer. I will guide you through the slide. On the very right side, we do have the authorities FDA and EMA deciding about cancer drug approvals. Next to it, we have the pharma companies who set up clinical trials to get approvals for developed drugs. The imaging CROs, or CROs in general, operationally support the clinical trials as a service provider for pharma, and in general, the so-called central read workflow with doubled blinded reads of images takes place here, of course, dependent on the trial protocol. But of course, cancer patients who participate in clinical trials are treated in hospitals, and there you can differentiate the job of radiologists as follows. First, they have the job to assess every cancer patient in clinical routine. We call it here the routine read. And all patients, participants of a clinical trial and non-participants of a clinical trial, get those reads. When patients participate in a clinical trial, they normally get a dedicated clinical trial site read where specifics of a trial have to be considered, like for example, the criteria conformity, like RECIST 1.1 or Lugano. Radiologists also work on own research projects for publications. Therefore, they also do research reads. MINT Lesion, the software we are providing, is currently used by different stakeholders and for different jobs to be done. Like in pharma, for imaging biomarker research, at CROs to conduct the central read workflow, but also at sites where we support all three use cases. The slide also gives an idea of our vision at MINT. Just 2 days ago, the pharma company Roche had a post on LinkedIn mentioning that 96% of oncological data are untapped because only 4% of patients are part of clinical trials. We would like to support you to make use of the remaining 96% routine patient data as well. Today's main focus of this webinar and for the upcoming RSNA is on the application of MINT at hospitals, cancer centers, but also radiological practices. If you have a major interest in the use case for CRO or pharma, please reach out to us and we will follow up after this webinar. Let's now make the deep dive and jump into the 3 different use cases, and we start with the routine read. The following slides are now built up in the very same way. On the left side, we have our current product offering. Next to it, we present the workflow, how MINT is applied for this use case. And below, we show what is new. The current offering for the routine use covers all templates to be applied in clinical routine, and this module will be automatically extended with all upcoming templates, as well as with all updated templates, meaning, for example, the upgrade from TNM7 to TNM8, or from Pirates version 2 to Pirates version 2.1. The workflow is normally as follows. Radiologists start the read in worklist, either in PACS or in the radiologist information system, jumps into PACS viewer, and in the moment he identifies an oncology-related job to be done, he jumps into MINT and applies a specific template. Further information and findings will be dictated into the existing reporting, but MINT auto-generate text blocks for the oncological part of the read, which will be integrated in the reporting as well. In addition, the MINT PDF with the holistic oncological disease overview is available and could be sent to PACS, EPIC, or also directly into the reporting. At RSNA, we highlight 3 areas with some substantial updates. We have again new templates in our module. We now also have the opportunity to upload existing external templates into MINT, and we made major progress again in the area of workflow and reporting integration. We now prepared a showcase using our latest rectum staging template, following the TNM8 guideline, as well as the ESGAR recommendation, and AJ will guide you through this.
Thanks, Tobias. So like you were just saying, MINT as a tool will guide the radiologist through the reading process and ensure criteria conformity and telling them what they need to measure, providing sophisticated tools, and generating these structured reports automatically. But not only is it, a guidance, it also allows the radiologist to do the assessment in a more explorative way. So it works just as a viewer, helping the radiologist determine where the disease is and using the different tools and methods possible to achieve the goal. So let's take a look at how this works for colorectal cancer. So as you can see here, as a radiologist, I would first go through the different series. I see that there is a tumor here, but I'm not sure if it's just one or perhaps there's multiple. So I'm going to go and look for the DCE image, and then MINT will synchronize this, and I can see now the wash-in and the washout, and this will then help me. So I can zoom in, take a closer look using the cursor, check the time intensity curve, and then after making my observation, I can conclude, for example, that this is in fact just one tumor. Then I will click on the plus sign in the primary tumor section. MINT will tell me to first classify the location. I say middle third. Then it now asks me to measure the size. It will prompt me with the right tools, take me to the right area, and then I can just go ahead and make my measurement as such. Once I'm done, there's several other things that MINT is now asking me to classify. So if I scroll down in my list, it's asking me, for example, morphology, if there is an anterior reflection. But of course, the next step would be to measure the craniocaudal length. I click on the section in the smart bar, MINT will prompt me with the right tool, and I can easily make my measurement. Similarly to the distance to the anocutaneous line can be done as that. I can also specify the circumferential location through this interactive diagram. And when I specify the depth of infiltration, for example, into the muscularis propria, then MINT will be able to use its algorithms to determine the T category. I can go on further to measure the minimum distance to the mesorectal fascia, as well as going down and determining the position where this minimum distance is present. And in this way, MINT is again telling me the MRF status is, in this case, currently free. I can go through the images to determine extramural vascular invasion and answer the questions regarding that in the application. Then I'm done for now. Let's say I want to see if there's some suspicious lymph nodes. I can go through my images to evaluate them. And then when I find one that I want to measure, I can use the regional lymph node category within the application using a bidimensional measurement to start my analysis. Once the measurement has been completed, MINT will ask me to specify a location and some characteristics like round shape, irregular border. Using this information, MINT will automatically determine if these lymph nodes were suspicious. Since that wasn't, let's take a look at another one which perhaps could be suspicious. Same procedure, I start with a bidimensional measurement, then I specify the location and the characteristics. In this case, if I answer it a little bit differently, you can see now that MINT is telling me that it is suspicious. And in this way, simultaneously calculating the N category. There's some other things I need to determine, for example, the ascites. So let's take a look at that really quickly. Using the relevant series that I need, I can make my observation and then go in and answer these questions respectively. I'm going to manually enter M0 for the metastases. And now let's take a look. We can see that MINT has calculated the stage. And if I hover over the TNM, it's telling me the explanation why it has done made the calculations in that way. There are some other details that I can go back and fill in. But once I'm done and I feel that I'm ready to approve the read, I just click on the button. Now, MINT is capturing all of this annotated information in a structured database. But of course, the report it is generating is also very useful for the treating physician. Here we can see on the first page, we have the overall assessment, a diagram for the lesions and the lymph nodes that were affected, as well as all the descriptions for the primary tumor and the regional lymph node. On the second page, I can already see some of the snapshots for the primary tumor that I measured, along with the area and length. Similarly, on the last page, there's the regional lymph nodes, again here with the snapshot and the size. And in this way, MINT is generating structured data and structured reports.
Thank you, AJ, for giving us an insight into the area of new templates. But we also have the areas of import of external templates and workflow and reporting integration. If you have a major interest in these areas, please come to visit us at our booth, and we will present you some showcases for those examples as well.
Moving on to the second area, or the second module of MINT, it is for the management of clinical trial site reads. So the core module contains all the relevant therapy response assessment criteria. So things like RECIST 1.1, iRECIST, Lugano, and so on. There's of course some additional add-ons like before for PET imaging, but there's a very interesting feature that we provide called the advanced criteria. With this feature, you can customize trials to fit a specific sponsored trial charter. So for example, if you have RECIST 1.1, but with some customizations to allow for more than 2 target lesions per organ, or something similar to Lugano but includes SUV or SUL measurements, MINT can model these rules into the respective clinical trial. The workflow here is a little bit different because we generally find that in a clinical trial site read, the workflow is in a secondary workflow. So what that means is MINT first is the primary tool for this assessment. So the radiology team would have a coordinator perhaps who would set up the model for the clinical trial with different trial arms if needed, and then will enroll the patients as the oncology team has provided the information for. Then the radiologist can go in and perform the baseline assessment and the follow-up assessments through the assistance of MINT lesion, and thus generating structured data longitudinally connected, and these reports which have tables, diagrams, and snapshots. MINT can also assist in the workflow and help communicate these results directly to the oncology automatically. This year at the RSNA, we're going to showcase some of the new criteria in this area as well that we've been working on. Another interesting topic that we're going to cover is the import of CDE trial forms. That is the Common Data Elements trial forms that can be automatically imported into the application. And finally, some of the enhancements we've done to provide excellence in the workflow. So like I mentioned before, in terms of communicating these results directly to the PI, or having the PI the ability to go into the application remotely and check the assessment as a view-only user and approve that or request changes if required. And in order to showcase this, let's take a look at how MINT can be used to import CDE trial forms.
Thank you very much, AJ, for this overview. Before we start to have a look at this showcase, you might have heard from the NIH CDE repository where you can browse and download CDE forms and single CDEs. The NCI, the National Cancer Institute, provides multiple templates to be applied in clinical trials. And we will now show you how you can directly import and use those templates and CDEs in MINT. Now, this is the repository I just mentioned, and you can click in and browse through the different forms. Here we have the area for NCI, and if you go through, you will find different templates. For example, one for eligibility for trials. So this is a standard template for eligibility. Each template has an ID, and you can easily paste this ID into MINT lesion, import the CDE, and MINT automatically takes over this template into MINT lesion. You can also do this not only with holistic templates, but also for single CDEs, meaning for single questions. For example, we can now integrate the question for the smoking history. Again, we take over the ID number that is available. We now select an existing template. We now select our lung cancer TNM8 template, and we just would like to use this template and add one specific question to it. For doing this, we are adding a new section. We are calling this section Smoking History. And in this section, we are now adding this single question and clicking on the button where we can now integrate a CDE and entering the ID. And we are having this question directly added to MINT. Now let's have a look on the showcase. First, we create a trial following now our new template. So our criteria that we are now using is the TNM8 enhanced with the smoking history question. Now we are looking for a patient that we would like to enroll. We select him and we assign him to the trial. What you also see that for this patient, we also have generated a routine report. I will come back to this later on. First step would be to fill out the eligibility template. So you just go in this general template provided by NCI, and you say, yes, it's eligible. And we also select the determined date where we did this decision. Now, we took over the information from clinical routine where we already had a lung staging and copied over the information into our trial. What you see now that you can directly use information from routine where we already did the TNM assessment with MINT with the primary tumor information, with the lymph node information, and with the distance metastasis. But in addition, in here, we have the smoking history question that we can directly fill out. Also, those questions are providing the information that are coming directly also from the templates, and you have it directly in your system. Of course, you can also get those data out of MINT with FHIR or other interfaces, and we are happy to show you this as well at our booth.
Yeah, so there's other criteria that we have developed, so we can discuss this if you have time to come by at our booth in RSNA, plus take a closer look at the different workflow excellence and the engines that we have, and how we can integrate this potentially into your workflow. We can have a closer look at that.
In the third area, imaging biomarker radiomics research, we offer one template which you can flexibly apply to all of your research projects. It allows you to skim radiomics parameters over time. Just to give you a short introduction into radiomics. Radiomics focuses on gray value information within a segmented area, for example, a lesion. First order analysis describes the distribution of the number of different gray values, and there are different parameters like skewness, kurtosis, etc., describing the curve. Second order analysis describes more the relation of gray values to their neighbors. Basis for this is a matrix showing how often a gray color is next to another gray color, for example, on the right side of it. If you have this matrix, a lot of parameters can be derived, which all are available in MINT-Lesion. AJ is now presenting you the showcase, and just have in mind that MINT could also provide an exported DICOM RT struct and or DICOM segmentation objects in addition to all these radiomics parameters.
So just keep in mind as we watch this video is that the radiomics research does not have to necessarily be done on a separate workflow. The beauty of MINT is that with the tools that we have, you can generate radiomics data and texture data as a byproduct of either routine reads or your clinical trial site reads. Of course, you can have dedicated workflows and dedicated reads to do specific radiomics research. And let's take a closer look at how that would work. So in this case, I have my research case, and I've done baseline and follow-up 3. So I'm going to jump into follow-up 4. I'm going to click on the findings that I need to follow up on. MINT will synchronize and give me the tool. It's a semi-automated tool where I just need to identify the lesion, and the application will automatically segment that in 3D. I do this for the 3 lesions that I have captured from baseline. Now we can see that if I hover my cursor over the individual values, I can see how they have changed longitudinally over the time points. There's also a histogram, which shows the texture. But if I click on the radiomics button, a whole different console now opens up. I can see all the first and second order parameters. I can highlight multiple of them to see how they progress in that graph in real time. Furthermore, I have the ability to change the resampling threshold and enable some different kinds of filters. I can go in and make different settings and changes to better understand this data and to analyze it from a different perspective. Then I can click on Calculate and generate this new data for myself. I can easily export this with a button on the right side to either XML or CSV. And it's not only a single patient that I can export, but I can export the entire patient cohort or my entire population. So in this way, you can have data coming in directly from your existing workflows or complemented by new workflows as well.
Thanks, AJ. Just to highlight this workflow as well and what AJ already mentioned, it's not necessarily that you do the radiomics in MINT just for your research. It's possible that you can take over that you captured in clinical routine or in your clinical trials to make your radiomics research. Just have this in mind. And if radiomics research is a big topic in your institutions, and maybe yourself are doing it or you have some colleagues doing it, pinpoint them to us. They should come by and visit us at RSNA to understand the opportunities of MINT regarding radiomics. Finally, we would like to spend the last minutes of this webinar on our AI approach. When considering the typical read workflow in radiology, there are multiple AI requests to make life much easier. Best case scenario would be that radiologists could say, I have support by automatism to find and classify findings. This is currently the major area where the most AI publications take place to automate lesion identification and segmentation and finding. And of course, this is a very important issue. But there are other things. Radiologists would like to say, I know all possible clinical consequences like pathways and tipping points. They would like to say, I have all images available to read the case properly. I know all relevant aspects of the patient history. I know the question to be answered to that are relevant for the referring clinician. And I have all related comparable cases available. To realize those requests, a combination of machine learning and knowledge management is reasonable. Not everything can be solved with machine learning. A good example are clinical pathways, which are very complex but could be modeled and applied within a software using knowledge modeling approach. Special about MINT is that we can apply those AI topics very context specific, since we know the context of each and every read. What we already have mentioned in the beginning, that we have the feedback loop between AI suggested and radiologist approved information due to the integrated reporting. Finally, we would like to highlight 3 different topics in the AI area. First, just to make sure that you got this idea of MINT, the MINT data are perfectly suited to train AI, like power food for AI. So if you plan to develop your own machine learning algorithms, MINT should be your data provider. Second, we would like to present you a showcase how we integrated an own developed algorithm into our workflow. That will be available very soon in our official release. And AJ will take over to present this part.
So this gets really exciting. Thinking of the possibilities of what AI can do to assist the radiologist. And one of the applications that we have come up with here at MINT is to allow the radiologist to identify one lesion at any given time point and let the AI automatically find and segment this lesion in the follow-up time points as well as the previous time points. So imagine you have a patient who's coming in with a lot of CDs or images on different disks. If you identify the lesion on one time point and allow the algorithm to run, it'll be much easier to detect how this lesion progressed over time in both directions. So let's take a look at how this would work. So in this case, I am in my follow-up 2 time point. I select my favorite semi-automated volumetric tool. I identify the lesion that I want to track, and then let the application automatically propagate this lesion forwards and backwards. So I can see down here how that lesion looked like at baseline and how it goes all the way up to follow-up 5. But remember that this tool is not just capturing the ROI, it's a volume tool that's also capturing a multitude of data, including texture parameters. And this is also where we're coming back to MINT is a cross-platform data engine where you can generate so much more data in the same workflow.
Thanks, AJ, for this showcase. Let's now also have a look into an example where we integrate an external machine learning algorithms, like an automated bone lesion detection in advanced prostate cancer. This tool was developed in cooperation with the Royal Marsden NHS Foundation Trust and the Institute of Cancer Research in London. The read starts with an already automated generated segmentation of bone lesions from a whole body diffusion weighted MRI. You can select now a single lesion and mark it, for example, as a target lesion in a bone red template. In addition, further image-based parameters are available, and the radiologist can add further attributes to the template that are requested, like you learned it already before in the rectum staging template or in all other templates we are providing. Of course, and as you're used it for MINT, all data are available to generate now visualizations to compare 2 time points and to generate a reporting which you can provide to your clinical colleagues. To summarize our AI approach, we would be very happy to discuss with you how MINT could be applied for specific use cases in your institution to be able to collect data over time that you can apply for own research, for own development of algorithms, but also to empower you to make the whole process in MINT much more efficient using knowledge modeling or machine learning applications.
With that, we come to the end of the presentation. First of all, we hope we really intrigued you and sparked your interest in what MINT can do in terms of transforming your clinical trial site reads, your radiomics research, as well as daily routine screening and staging applications. We are very happy to have 2 separate exhibitions at the RSNA this year. Our main exhibition will be in the North Hall, Level 3, booth number 8349A. Here you can meet with us and the entire team, take a look at these showcases again in more detail, and have more detailed discussions about what we can do for your center. But in addition to this, we will have an AI showcase in the North Hall Level 2, booth 11630. Here we can take a look in more detail specifically at AI-related topics, and on Thursday, the 5th of December at 1:30 p.m., we will have a presentation in the AI Theater. Lastly, we were also going to be hosting an evening reception on the evening of on Wednesday, where you can have drinks with us, meet some existing customers, and enjoy the night. With that, we want to thank you for your attention and your time. We hope you enjoyed this presentation. Please leave us your questions, and we will send you an email with the answers, as well as a link to the recording of today's presentation. Thank you and good night.